So what I would like to do is to tell you about, of course, the work that we do on human cortical neurons, and tell you how they may relate to human cognition. Of course, a lot of that, let's say, relation between neurons, their activity, their function, and cognition comes from work on rodents, and of course, and other laboratory animals, also on monkeys. And of course, laboratory animals and monkeys and other species can show amazing behavior, but of course, it's very different from human cognition. So, for instance, to give you an example, of course, our closest relative, the chimpanzee, uses tools to, for instance, get to its food. And when you have in a laboratory setting, chimpanzees can do amazing things. For instance, Ayumu, as this one is called, can memorize 9 locations in that specific order in a flash. The screen is flashed for, let's say, less than a second. He immediately sees the order of the numbers, and then when the screen is off again, he can immediately reproduce the pattern of those numbers instantly. So if you try to do this yourself for the first time, it's really hard. I mean, I cannot, I mean, maybe 3 or 4 locations, and then I'm lost. But he can do it all 9 and over and over and over again. And that's a bit of the trick, of course. You need to train. He has been training for years to be able to do that. Of course, so that shows. I mean, chimpanzees have a wonderful working memory, especially when they are trained. But of course, that's not the same as the type of mental ability that humans show, and especially, of course, we are interested in science. So, of course, the things that scientists do, of course, is amazing. But also in other fields, there are amazing things that humans do, that the human brain produces, that we cannot study in laboratory animals. So the question that drives that we focus on today is whether we can pinpoint neurobiological factors that are driving factors for human mental ability. Of course, and then the big thing is, of course, we can speculate about that. We can drink a glass of wine and speculate, well, we have a large brain and that's why we are so smart. But can we actually test what those neurobiological factors are that may contribute to human mental ability? So, of course, the large brain was already mentioned. We have a large brain. We have a large neocortex that is folded. Of course, when you look at this coronal view, you see that neocortex has a much larger surface than in other animals, and particularly than the mouse. And also, when you look at the number of neurons in the cerebral cortex plotted against the mass of the cerebral cortex, then we see that we have a lot of neurons in our brain. But actually, they're in our neocortex, but actually this whale has twice the number of neurons in its neocortex than we have. So it's not only, so we have about 19 billion neurons in our neocortex. This whale has about 40 billion neurons in its neocortex. That's in this Mortensen et al. Paper. So maybe neurons, the number of neurons is not the only factor that drives mental ability. Perhaps it's the properties of neurons that drives that. So, and there's another thing, when you look at the layers, we already talked about layer 2-3 a lot. And when you compare that with the layers in other monkeys, in this case, then you see that human neocortex has expanded, and it's not only thicker, but especially layers 2 and 3 have expanded more than the other layers. And when you look at it in a relative fashion, so this is in absolute numbers, you see also compared to other animals like rats and cats, the human neocortex is thicker, but layers 2 and 3 in particular are thicker. And when you look at it in a relative sense, you see that in humans, layer 2, 3 occupy much more of the neocortex than the other layers compared to other animal species. Now, the big question is, of course, is this relevant for human cognition? And can we test this? So the way we approach this is by starting to work with neurosurgeons. At the field was already mentioned by Elina. We get tissue from either epilepsy or tumor patients. When we get tissue from epilepsy patients, so epilepsy surgery, not everybody realizes, during epilepsy surgery, there is a lot of tissue taken away by the surgeon. Sometimes it's 4 or 5 centimeters that they take out from this pole with the temporal lobe. When we go in, the surgeon first cuts out a cubic centimeter of tissue right in the middle of the area that he's going to cut out, that she's going to cut out completely, and we quickly take that to the lab to be able to record some of the neurons, reconstruct them. Elina showed that beautifully, but also do some cortical structure analysis. We analyze RNA expression of each of many neurons, single neurons. We get MRI data from the same patient, but the patients also go through a series of cognitive tests before they go into surgery, so that the psychologist can estimate the baseline performance on cognitive tasks that have to do with this brain area. So like attention, short-term memory, long-term memory, vocabulary, and all those things, but also IQ tests. And I'll come back to that in a second. They also take IQ tests. And the reason is that once the surgeon has taken out this tissue, the patient goes back to the psychologist and does the test again, so that the psychologist can determine what kind of function loss has occurred and whether there are specific therapies that can retrain that property of the patient specifically. So, so these data are also of the same patient. So we get a multimodal data set of the same subject, and that we then can hopefully, if we get many subjects, relate different properties to their cognitive functioning. Now, the patients also take IQ tests because this area has been implicated in IQ. So there are many studies on healthy college students where they would fill out an IQ test and then go into the MRI scanner and have a structural and functional MRI taken. This is reviewed in this paper. And what they came up with is that there are certain anatomical features that correlate with the IQ scores of those students. And in particular, area 21, the thickness of area 21 associates with IQ scores positively. So the higher the IQ score, the thicker temporal, medial temporal lobe, medial temporal gyrus is. Now, of course, we know that, of course, on the left side of our brain, the language centers are located. So this whole system is involved in language, but it's not entirely clear what area 21 does. So at least this is the only real, let's say, the closest to what you get to a functional readout of what this area is doing. Now, in the IQ tests that are being taken, this is the adult Wechsler scales, or the WISE 3 or 4. There, this is a standard IQ test that has been developed over many years. There are several numbers that come out. So the test itself consists of different parts. So there's verbal IQ, working memory, the perceptual organization, and processing speed. Those are categories that are being tested. It takes about 2 to 3 hours to fill out this IQ test. So it's really a serious test. And then a couple of numbers are generated that relate to verbal IQ score and the performance IQ score. And there's one number that reflects the full scale IQ score. So I will use verbal IQ and full scale IQ sometimes mixed, but you will see it from the graphs. Now, the first question is, do all cortical layers contribute to the association between IQ and cortical thickness? So, of course, to test whether layer 2-3 has any functional consequence for cognition, let's look at whether all those layers, whether the thickness of layers is relevant for cognition. So what we do, and similar to what Javier showed, we get tissue to the lab from the surgery that can be this tip of the crown of the gyrus. And of course, cortical thickness varies when you go along that crown. So what we did is we quantified along the crown the thickness of the different layers, and looked at patients with high IQ scores and low IQ scores at the thicknesses of the different layers. And basically, what we found is that when what really stuck out, it was remained statistically significant when we corrected for multiple testing, was the thickness of layer 2-3 on the left side, and this is the correlation with verbal IQ. See, there's low verbal IQ, high verbal IQ, and you see that in patients with low verbal IQ, they have a lower thickness, or a thinner cortex, than patients with high IQ. Of course, in healthy subjects, the left temporal cortex is layer 2-3 is always thinner than the right temporal cortex. So in the patients with high IQ have a thicker temporal cortex layer 2-3. And when we correlate, that verbal IQ score with left cortical layer 2-3 thickness, you see that there's a strong positive correlation. So what does this cortical thickness affect cellular architecture? We've been discussing this also a couple of times today, but I wanted to show you for different subjects how cortical thickness relates to the architecture of the cells inside that tissue. So again, we quantify, we stain, and you see all the cell bodies, and then we quantify not only the density, but also the cell sizes and stuff like that. And correlated that with the thickness of the cortex of the different patients. So here you see two examples, and here you see that the thicker the cortex is of that medial temporal cortex, the lower the density of the cells. So these big dots are individual patients. And you see that with the increasing thickness, the cell bodies become larger. So even between humans, there's variability, and it seems that cell sizes and densities vary between subjects, and with thicker cortex, lower density, larger neurons. So what type of pyramidal cells are there? So this paper has also been discussed already with what we did together with the people from the Allen Institute and with Karo Tamás in Hungary. We recorded from neurons taking physiological profiles, as Elena explained, also reconstructed the morphologies, but also got the nucleus out and had the Allen Institute analyze the RNA content of the nuclei of these human neurons, and thereby being able to not only get the transcriptomic landscape of all the cell types, but also their location within the cortex, compare them to the mouse cell types, and of course get the full reconstructions and physiology. And of course, the interesting part was that the deep layers pyramidal neurons, the large ones, they have no mouse homologs, and they are particularly vulnerable in Alzheimer's disease. So this is an interesting population of neurons, and they are large. They have large morphologies, as you know, these layer 3 neurons. So the next question is neuron size related to cognitive ability? So again, we looked at low IQ versus high IQ patients, reconstructed cells in their temporal cortex. And the first thing that we did, as what Natalia did with a couple of internship students, was to just measure the cell body diameter, basically like this. And then we did that because we had many cells from the patients, but we did not get full morphologies from every cell. So some cells were truncated, but of course we still had the cell body. And what you already see from this analysis is that the cell body diameter in patients with high IQ scores are shifted to the right in the distribution than the cell body diameter of patients with low IQ scores. But when we looked at the full reconstructed cells, which is plotted here, and we plotted their total dendritic length against IQ scores, then we see indeed a positive correlation between that total dendritic length and IQ score. So the larger the IQ score the patient has, also those patients tend to have cells with longer dendrites and larger cell bodies. Now, of course, we know already that from work from Chris, but also from others, that of course the human dendrites, the total dendritic length, are larger than, for instance, mouse and macaque neurons in layer 2-3. But also we know from Javier's work in the past that layer 2-3 neurons receive many more synapses than mouse and rat neurons do in layer 2-3. So there's almost double the amount of synapses that layer 2-3 neurons receive. And so the question is, what about synapse function? How do these synapses, what do they look like when you stimulate them? So we started recording. So this is a paper by Chris and Sarah Hunt that was done in collaboration, of course, with the Blue Brain. Paired recordings but we did also for pair recording in the past, where we looked at other aspects, where Chris observed and Sarah observed that these human synapses, pyramidal to pyramidal synapses, unitary synapses, have a large amplitude, and they are also much more reliable, so they show lower failure rates than mouse synapses between pyramidal neurons. What we also observed is that human glutamatergic synapses, although they are depressing, they have a fast recovery from depression. When you look at the amplitude of this synaptic response 500 milliseconds after the train, you see that the amplitude is already back to the first EPSP in the train, but in the mouse synapse, you see that it is still depressed. So the recovery rate has a faster time constant, a lower time constant in human synapses than in mouse and young mouse and adult mouse synapses. And this was basically also shown in the Campagnolo. These two things, so strongly reliable synapses and fast recovery from depression, were also one of the main conclusions of these glutamatergic synapses in that paper. So this has consequences for information transfer between two pyramidal neurons, and that's what Michele Giuliano quantified using the models that were developed in this paper by Ivan Segev and Misha Tsodix, and calculate and using those models and only changing the recovery rate from depression. Is what they found is that between those synapses that have a slow rate of recovery from depression and a fast rate of recovery from depression, you see that with increasing firing rate, these synapses that recover fast from depression can transfer much more information than synapses that have a slow recovery from depression. So there's per synapse, there's much more information transfer between pyramidal neurons. So they also receive many more synapses, as I already told you. And the question is, so there's a lot of information incoming into these dendrites, reliable synapses that are stronger, that recover fast from depression, and thereby transfer much more information, but also many more synapses because the dendrites are larger. So the question is, can human pyramidal neurons actually process that kind of synaptic inputs into action potential output, and then in particular action potential output timing? So that's what we tested together with Guilherme. So we did an experiment that was inspired by Michele Giuliano's work, where what we did is we played an artificial input in the membrane, subthreshold membrane potential changes, a sine wave, and then we depolarized the neuron a little bit so that it started to fire action potentials at a constant rate, somewhere between 5 and 15 hertz. And then we would look whether the neuron would time its action potential to the sine wave that we were playing in the membrane subthreshold membrane potentials. And this is what we found for in the human neurons. When we play a frequency of 10 hertz, then we see that the action potential timing tends to be at the peak of the sine wave. When we played a sine wave of 100 hertz, the timing was a bit shifted to the membrane potential changes, but it was still phase locked to the sine wave. But even when we played 100 hertz, so we varied the membrane potential at 100 hertz, at 1000 hertz, the action potentials would be timed relative to the peak, to the sine wave that was artificially put on the membrane potential. So it's not that the neuron starts to fire at 1000 hertz. No, where it fired at between 5 to 15 hertz, but when it fired an action potential, it was in relation to this sine wave. And this is particular for human neurons because mouse neurons or rat neurons, in this case, from the study of Michele Giuliano, they don't follow these frequencies. So action potential timing is lost with frequencies beyond 200 hertz. So but the human neurons, they continue to time their action potential relative to that subthreshold membrane potential change up to 1000 hertz. We even sometimes tested higher frequencies. We could never, the neuron was always following those frequencies, time locking, phase locking its action potential timing to that sine wave. So apparently they can encode a much higher bandwidth of input signals into their output by just timing that. And you have to realize, of course, that this is for only one neuron, if you would have a whole network of neurons, so Alida was explaining that the dendritic complexity results in much better computational properties, memory encoding properties. So yeah, of course, also in information processing, you have to think in network terms. So if each neuron is able to time its action potential to a much higher bandwidth input to the network, then in total, you will get a reflection of a much richer repertoire of inputs than with these slow neurons. Now, the big question, of course, there's an interesting phenomenon that's nice to observe, but what is the mechanism that underlies it? So, of course, from theoretical work, we know that the exponential rise speed, onset rapidity is very important for this kind of encoding of exponential timing. You can imagine that with a slow exponential, when there's a fast synaptic input, the neuron would just miss the reporting that slow input, or that a fast synaptic input with a slow exponential. But with a fast exponential, the neuron would be able to reliably report when that input arrived. And what Guillermo observed was that human neurons actually have a very fast exponential rise speed. Whether you have a single exponential, it's not that different from a mouse neuron, but especially when the neuron starts to fire repeatedly at higher frequencies, and you look at the derivative of the membrane potential change, you see that the mouse exponential slows down a lot, and will not be able to reliably, especially at these frequencies of 15 hertz or so, will not be able to reliably report when a fast input arrived on its dendrites. And thereby, a slow exponential will have a much smaller bandwidth of reporting input frequency content in its exponential output timing than a fast exponential would have. Now, but of course, we can all. This is all very interesting, but how does this relate to human cognition? Does this actually have any impact on human cognition? So the question is, does exponential speed relate to human cognition? So we did again the same thing. We looked at neurons of patients with low IQ and high IQ scores and quantified their exponential kinetics, the onset rapidities, with also rise speeds at different frequencies, single exponential, but also 0 to 10 hertz, 11 to 20 hertz, etc. And quantified that for high IQ scores versus low IQ scores. And what we see is that, in particular, with repeated exponential firing, the exponentials in patients with low IQ scores, in the neurons of patients with low IQ scores, slow down much more than the exponentials of patients with high IQ scores. And when we look at the correlation, then we indeed see that there's a positive correlation between exponential stability or rise speed stability at higher frequencies with higher IQ scores. It is as if with fast exponentials and with repeated firing, patients with high IQ scores, their neurons can keep on encoding information into their exponential outputs much better than neurons with slow exponentials in patients with low IQ scores. Now, what is the mechanism that underlies this fast exponential initiation mechanism? So there are several proposals for how the fast exponentials can be initiated. There have been hypotheses about exponential sodium channel cooperativity. The sodium channels would somehow cooperatively open and thereby speed up the activation. So we thought, let's just start to record sodium currents and potassium currents in human neurons and subject them to repeated activity just to look at the kinetics and stability and voltage dependence from humans and neurons repeatedly activated in nucleated patches where we played exponential command traces, voltage command traces. We recorded their sodium currents and their potassium currents at repeated firing. This is the first exponential, fifth exponential, 200th exponential, and then look at the amplitude of the sodium current and the amplitude of the potassium current. And you see that both in human and in mouse, the sodium currents, they reduce in amplitude. Also, the potassium currents reduce in amplitude, but in mouse neurons, they reduce much more than in human neurons. And that is quantified here. So you see that even at the second exponential already, there's a big change in human neurons, and a little bit more down the road. Potassium channels are a bit more resistant to reduction, so it takes a bit before they actually become significantly different between human and mouse. But they're definitely human potassium and sodium channels are more resistant to say desensitization than mouse currents are. So are the voltage dependence and current kinetics different? So we looked at the steady state voltage dependence of, in this case, sodium channels, and we see that human sodium channels are right shifted in their voltage dependence, so their inactivation curve and their activation curve are right shifted, but also the kinetics of inactivation are different. So there's a slower kinetics for inactivation in human sodium currents at all voltages, and there's a faster recovery from inactivation in human sodium channels compared to mouse. So all in all, so a higher voltage dependence, dependence at higher voltages, slower inactivation, faster recovery from inactivation. That means that the sodium channels are more available in human neurons for activation. And to systematically address which properties are responsible for the stability of the sodium current, we made a simple Hodgkin-Huxley model of these currents, and we started to replace mouse sodium channels with different human properties. So activation properties of human channels, putting them into mouse channels that didn't change the stability of the sodium current. So you see the normalized sodium current amplitude at the 5th exponential. That didn't change much, but when we started to give mouse sodium channels human inactivation properties, here's the steady state inactivation, the time constant of inactivation, then you see that the mouse sodium channels start to become human-like and have the same type of resistance to desensitization. And also when we simulated this same experiment in the model, then we see that replacing mouse sodium channels with human sodium channels, that shifts the bandwidth of exponential timing to these input frequencies to the right, so to higher frequencies, suggesting that indeed these sodium channels contribute at least to this property of higher encoding capacity of human neurons. So that brings me to the first intermediate conclusions, that we've seen that the structure and size of neurons, structure of the cortex and the size of neurons associated with cognitive ability. There's a large synaptic information transfer in human neurons. Human sodium channel function enables stable action potential kinetics, providing a larger bandwidth for encoding. Oh, that was it. Sorry, that was it. So, so in all, it's these channels, but also the protection channels that contribute to this stability of the large bandwidth encoding. So, of course, we've been talking a lot about pyramidal neurons today, and the need for more detailed information on interneurons. We totally agree. So, together with the Allen Institute, we have submitted, and with Chris and with Gabor Tamas, we have submitted a couple of papers recently there on bioRxiv that really, let's say, make a nice inventory of triple modality properties of interneurons throughout the layers, also in layer 1. So you can find those on bioRxiv. What I wanted to tell you about is a more detailed story about a particular type of interneuron, the fast spiking interneuron. And what we were wondering is we were struck by this property of brains, of mammalian brains, and that is when you look at small brains or large mammalian brains, that many of the rhythms that can be recorded in EEG actually are stable in their frequency. So when you look at the gamma frequencies or alpha or the slow frequencies in the delta range, let's say, they have pretty much the same values. Only the theta frequency is the one that really falls off with a larger brain size, but the others are pretty constant. And in particular, these fast rhythms, they seem to be preserved evolutionarily. At the same time, we know that, of course, the parameters of neuron densities, neuron sizes, etc., are very different between small brains and large brains. There's much lower density, much larger neurons, distances between neurons are much larger, the distances between brain areas are much larger. So the question that we had is, well, how can this, let's say, this basic circuit motif that underlies gamma frequencies, where you have a pyramidal neuron to interneuron connection, and then an interneuron to pyramidal neuron, either back or to the next pyramidal neuron connection, fast spiking interneuron. How can that, let's say, what is evolved in this human circuit motif that allows the brain to have a similar frequency of gamma rhythms as in the mouse brain? Or to draw an analogy, if you take a train from, well, let's say from this place to Winterthur, I think this is, it's about 260 kilometers. It will take you about, well, 3 hours and 15 minutes. Yeah, that's interesting, but you may find that your friend is also taking the train from Amsterdam to Paris, which is 520 kilometers, and takes the same amount of time to get there. So it travels a much larger distance. Of course, we know there are all kinds of adaptations to make this happen. So either in the trains or in the tracks that allow for a much larger speed. So the question is, what happened in the human brain that was optimizing speed over larger distances, in particular in this fast spiking interneuron circuitry motif that allows these for this conservation of evolutionary conservation of gamma rhythm. So what we did basically is what we wanted to do. We wanted to solve this the timetable of traveling of an action potential from the human pyramidal neuron to the synapse on the fast spiking neuron, then to the cell body, generate an action potential, and then initiating an IPSP or inhibitory postsynaptic potential in the next pyramidal neuron, and compare that and look at the timetables both in human and in mouse, and what kind of structural and functional adaptations have been optimized to do that. This project was done by René and Anna, Stan, and Tim in the lab, particularly. And the first question is, well, let's have a closer look at the dendrites of fast spiking interneurons. Are they different? So this was these are the papers that I mentioned that we've put on bioRxiv, or they have put on bioRxiv. Gives a large inventory on both transcriptome, EFIS, and morphology of interneuron types across the human temporal cortex. But we are going to focus only now on one type, the parvalbumin positive type. This is the human. It also includes axons, of course. And we are going to look at the dendrites of these human fast spiking neurons versus mouse fast spiking neurons. And René quantified those. And what we observed is that the total dendritic length indeed is a bit larger than in mouse. That the number of branch points is actually reduced in human fast spiking neurons, so they're a little bit less complex. But and at the same time, also the number of dendrites that come out of the cell body is also lower in humans. So the mouse fast spiking neurons have more dendrites coming from their cell body. But does this affect EPSP propagation? So we made a model of the fast spiking interneuron. The model fired action potentials like you would expect, very stable, and put synapses on different locations on these dendrites, both in mouse and in human, and then compared EPSP properties in these models, where the synapse to soma path length was changed for equally in the different models. So what we systematically observed was that the human EPSP or the EPSP plate or inputted on the human morphology was always smaller than on the mouse morphology. And this was for all path lengths, this was the case. So a similar input with the larger morphology would just generate lower synaptic amplitude. So are EPSP smaller in humans fast spiking interneurons? So we started to record this. Paired recordings is what René did, generate a spike, and what we observed is that actually the amplitude of the excitatory input to the fast spiking interneurons is not different. There's no difference with any amplitude. So can we explain that difference in the model? So how can we change the morphology in such a way that we can explain this? And what we see is that that when we again do such a, let's say, a replacement strategy in the model, where we may have a human model that has a certain EPSP amplitude, and then replace the path length with a mouse path length, that immediately increases the EPSP amplitude. So, and when we change, and when we have a mouse model and we change the path length to a human model, then it immediately decreases the EPSP amplitude. And then we changed let's say we made the human model, we changed the dendrite count, so the number of dendrites that come from the cell body, changed that to a mouse neuron. That we immediately decreased the EPSP amplitude, so we reduced the number of dendrites coming from the cell body. And when we increased the number of increased the number of dendrites coming from the cell body of the mouse neuron, that when we decreased that increased the EPSP amplitude. What also helped was if we increased synapse strength of the synapse coming onto the dendrite by 2. That will also generate a larger amplitude. And actually, from EM work that Gabor Tamas published, he showed that the excitatory synapse from the pyramidal neuron to fast-spiking interneuron actually is much larger. Has a much larger active zone, has many more vesicles docked there, and has a much larger amplitude than the rodent synapse. So we think that a stronger input is justified, and so then both the dendrite adjustments, so longer dendrites with less complex and less coming out of the soma, and a stronger input will explain that these synapses in mouse and human fast-spiking interneurons have a similar amplitude. So is the fast-spiking output different? So what we first did is we looked at what kind of action potentials the model generated. So we again modeled the different morphologies, and then have the model generate action potentials. And what we see is that that because there's a much larger dendrite connected to this fast-spiking cell body in human morphologies, the onset rapidity is faster, and the AP rise speed is faster, and the fall speed also. So the kinetics of the action potential is different in the human fast-spiking interneuron models than in the mouse fast-spiking interneuron models. And we just again replaced the size of the dendrite, just like we did in the paper with Guy Ayal in Idan's lab. And then we saw that the dendritic load on the cell body will determine what the onset rapidity is of the action potential in the actual initial segment, so the timing thereof, and in the cell bodies. So the bigger dendrite will just generate faster action potential onsets. So can human fast-spiking interneurons generate fast synaptic responses in the pyramidal cell? So we go to the next station, the connection between the fast-spiking interneuron and the pyramidal neuron. And record it now again in slices, single unitary connections between fast-spiking interneurons and pyramidal neurons, and activated them like this. And what we basically observed is that that the human synapses tend to have a much faster, shorter delay between the action potential and the onset of the inhibitory input in the pyramidal neuron than that the mouse unitary synapses have. They tend to be a bit slower than human synapses. And this is quantified here. So the onset latency is shorter in human synapses than in the mouse synapses. And the amplitudes are also different. So there's a bit stronger amplitude in the human synapse than in the mouse synapse. So that's now we can fill in the timetable of an action potential information traveling through this circuit, and we find that both human and mouse, these numbers, when you fill them in, add up to a very similar timetable of 3.5 milliseconds, going from an action potential here in the presynaptic pyramidal neuron through the interneuron to the next pyramidal neuron, or back to the same pyramidal neuron. So we think that these are the kind of adaptations that we find in this circuitry. So the dendrites are longer, but the structure is more simple, optimized for fast conversion. There are lower number of dendrites coming out of the cell body, and they are longer and less complex. the delay, synaptic delay, are between the pyramidal and the fast spiking neuron are similar, but the synapses are stronger. Action and initiation kinetics are faster, and the output of human fast synaptic neurons to the pyramidals is also faster. All right. So again, for the third part, the last part, I'm gonna, if you bear with me, I'm gonna switch gears a little bit, because we're going back to neurobiological correlates of human mental ability, human intelligence. So basically, that field is split between two approaches. So you have the brain imaging approach I told you already about. So there are brain areas that are correlated or associated with IQ scores in healthy subjects. And at the same time, at the other end of the spectrum, there are large genetic studies, genome-wide association studies that identify genetic variability in regions or SNPs in the DNA that associate with IQ scores. And so these studies, they will give us areas, brain areas of intelligence, and these approaches will give us genes associated with intelligence. So different studies give different numbers of genes, but there are hundreds of genes that have been associated with genes with IQ scores in healthy subjects. Now, our goal is, what we want to understand is how these genes, since they express in neurons and give certain properties to neurons, large dendrites, fast action potentials, stuff like that, that results in properties of circuits and ultimately properties of brain areas that give rise to these associations in brain imaging. We want to understand how this is related to each other. So what we have been doing this is what we have been doing in several studies, but the next question that we have is, well, since we have those neurons that and their properties that are associated with IQ scores, can we now look at the genes that have been associated with intelligence, whether there are genes that are expressed more in those neurons, in those large pyramidal neurons, and can we then understand how these genes may give rise to this type of function of these neurons, fast direction potential, larger dendrites. And at the same time, of course, we have now access, and with all these recent fast developments in RNA sequencing data, we have access to single cell expression data, and also from our own Patch-seq data that we collected together with the Allen Institute and with Gabor Tamas. We have, for each of those neurons, we have an expression profile of which genes they express. So the question is, do the neurons that we recorded and the neurons that we identified, different cell types, do they express genes of intelligence more? And that's what you see here. So what Natalia and her team did with Jai Heyer and Stan Driesens and Anna Galakova, they calculated, looked at the mRNA expression for the genes that have been associated with intelligence and looked whether they express more of these, they have higher levels of expression of these genes. And it seems that these, especially these large types that are located deep in the temporal cortex, they tend to have more of these genes expressed. But do these genes and their expression levels, do they correlate with the neuronal properties that we identified that associated with IQ scores? So we've associated dendritic length with IQ scores. You see that the different types have different total dendritic lengths. But when we then correlated the level of gene expression, here color-coded, with the size of the neurons, here is total dendritic length. The neurons are ranked by total dendritic length here on the in the rows, and this is the rows are in the columns. There are the neurons, of course, and their sizes, and then in the rows you see that each of the row is a different gene that has been linked to intelligence. You see the expression levels, so the mRNA levels of these genes coded in color. You see that there are genes that positively correlate with dendrite, and there are genes that negatively correlate with dendrite size. So some genes that have been associated with intelligence from GWASs have a positive correlation with large neurons, are more expressed in large neurons, and some of the genes that have been associated with intelligence tend to be more expressed in the small neurons, and that is quantified here. And the same we did for the action potential speed. So we've identified that action potential rise speed and the stability thereof is associated with IQ scores. And you see that the different neuron types have different action potential stability, different action potential rise speeds and stability. You see those, especially this type, the CARM, has fast action potentials. And we did the same thing. So we looked at the genes. Each row is a different gene. Now we ranked the neurons according to their action potential speed and stability. You see that again that there are some genes that are positively correlated in their expression in fast neurons, and some genes are negatively correlated in their expression and are more expressed in neurons with slower action potentials. Now, of course, we're going to look at what type of genes those are. But first, what we did is we not only looked at the IQ genes, but IQ scores also correlate strongly with educational attainment. And there have been, so educational attainment means how long have you been going to school and what level of education did you reach. There have been GWASs on that have identified genes. And there's overlap between IQ genes and educational attainment genes. But there are also these genes that are located in human accelerated regions. These are regions of the genome that have a faster evolutionary speed or mutation rate compared to chimpanzees and other great apes. Then in humans, a faster evolutionary speed than other regions, and there are a number of genes in there, and there's also overlap with the educational attainment genes and the IQ genes. So when you look at the number of human genes that are located in human accelerated regions, so humans that regions that show fast evolutionary changes, then you see that in the educational attainment gene group, this overlap is about 19%. And for IQ genes, about 14%. But when we look at the same thing, but then only with the genes that correlate with total neural length and IP speed that are expressed in those pyramidal neurons, then you see that there about half of those genes are located in human accelerated regions. So these are apparently genes that drive certain aspects of human evolution. At least that's the hypothesis, of course. So what kind of genes are those? So we did gene ontology analysis, the genes that correlate with total length and fast action potentials. When you do gene ontology, that means you look at which processes they are overrepresented in. And there's all kinds of synaptic processes. There's dendritic development, synaptic signaling, or ion regulation. But we've listed those genes here, and I'll highlight a couple. And there are many receptors and ion channel related genes. So this is a calcium channel subunit, the 2D delta, the 2D3 subunit. These are two glutamatergic, metabotropic glutamatergic receptors, serotonin receptor. These are two. This is a potassium channel subunit. This is a potassium channel interacting protein. It actually binds potassium channels and calcium channels and sodium channels together. And this RIMS1 is a presynaptic gene that gives a lot of the synaptic glutamatergic release properties. So, and when you do a synaptic ontology, then you see that many of the genes are also involved in processes at synapses. So we think that these kind of genes can point us, can lead our hypotheses guidance towards figuring out how we can understand how these kind of genes contribute to neuron function and why they are associated with human mental ability in the GWASs. So that's one of the next steps that we are taking is we're going to manipulate expression of these genes in human neurons, because we are culturing the neurons or phenotypic slice cultures, and we will manipulate expression of these genes to see how they contribute. To end the talk, of course, what I've shown you, is we think that there's an importance of the size of the brain, the amount of brain we have per body mass, frontal areas, and more neurons in cortex for the evolution of mental ability. Of course, humans have bigger brains, but we think that properties of neurons and synapses, like large dendrites, more synapses per neuron, more information transfer per synapse, faster action potentials to encode those synaptic inputs, that actually drives mental ability much faster. because there are so many neurons and so many synapses in the brain. If you increase the computational power of a synapse, they do it across the brain, that increases computational power much more. So you have to really think of the evolution of mental ability like this. Brain anatomy is driving, of course, increase in human mental ability, but changing properties of dendrites, synapses, information transfer, extra potentials, and other cell features, because that will speed up this development of human mental ability tremendously, and will also make us better understand why the difference between human brains and brains of other species leads to such a difference in mental ability. And we didn't even talk about other cell types yet, and other features yet, because of course there's a whole world to discover in the glia cells in human brain, because human cortex has two-thirds of the cell types are glia cells, only 1/3 is neurons. In the mouse brain, it's the other way around. It's one-third is glia cells, 2/3 is neurons. So I think there's still a lot to discover. Anyway, I want to thank our collaborators. The people that did the work in the lab, of course, Natalia, Christian, and all the PhD students, of course, the neurosurgeons, the people in the Allen Institute collaboration, Gabor, of course, for his support and inspiration, and of course the people that make the models, because then we actually understand what we are measuring. So I want to thank them also very much, and of course the funders, and I would like to thank you for your attention.